Good afternoon, everyone. I would like to welcome you to the second webinar of the Quality Webinar series organized by EATRIS. My name is Martin de Kort. I'm Scientific Program Manager at EATRIS, and I will be hosting this webinar today. Today, we are very pleased to have 3 speakers joining this webinar on quality in high-throughput screening. Before I introduce our speakers, I want to make you aware that this webinar is being recorded, including the questions and answers at the end of this session. It will be made available on the EATRIS website shortly after the finish. We have reserved some time at the end of this session for questions, and we invite you to write down your questions in the question box in the control panel, as you can see on this slide. The first speaker of today is Matthew Hall. Matthew Hall is Biology Group Leader at the Division of Preclinical Innovation at the NCATS Chemical Genomics Center, where he develops and optimizes biochemical and cell-based assays for automated small molecule high-throughput screening. His research focuses on oncology, but also covers a diverse range of other human pathologies and diseases. Prior to joining NCATS, Matthew was awarded a fellowship at Johns Hopkins Bloomberg School of Public Health, where he investigated the cellular regulation of transition metals such as manganese. He then moved to the NCI lab of cell biology, bringing together his chemical and genetic experience to work on cancer multi-drug resistance. He ultimately became a staff scientist and led a number of programs that included developing imaging probes for drug transporters at the blood-brain barrier, understanding mechanisms of resistance to cisplatin, and understanding the phenomenon of collateral sensitivity in multi-drug resistant cancer cells. Dr. Matthew Hall obtained his PhD in cell biology and medicinal chemistry at the University of Sydney. He has published over 70 peer-reviewed papers and is on the editorial board of Drugs of the Future. The second speaker of today is Wolfgang Fecke. Wolfgang obtained his PhD in '94 at the Heinrich Heine University in Düsseldorf on the function of protein complexes in mitochondria. After a postdoc at Columbia University, he worked as group leader in several pharmaceutical and biotech companies in the UK, Germany, and Italy on drug discovery programs, always with a focus on assay development, screening, and lead profiling. In 2015, he helped to establish the Translational Discovery Sciences Group at the VIB Institute in Leuven, in Belgium. Wolfgang returned to Germany in 2018, becoming director of the new European infrastructure, EU-OPENSCREEN, in Berlin. Third and final speaker of today is Martin Neuenschwander. Martin studied biochemistry at the ETH in Zurich, in Switzerland. In 2006, he obtained his PhD at the Institute of Applied Biosciences of the ETH Zurich with Donald Hilbert on the development of enzymatic high-throughput assays in living cells. He then worked on the automation of lab processes for directed enzyme evolution using liquid handling workstations. In 2008, he joined the steering core facility at FMP in Berlin in Germany, and his work encompasses the setup of lab automation for compound handling and screening, software development for data analysis, and project management for screening campaigns. I will now hand over to the first speaker, Matthew.
Wonderful, thanks everyone. So I'm going to talk a little bit about quality and high throughput screening today. It's obviously a deep and complicated problem. I'm going to tell you a tiny bit about what we do at NCATS to give you some context from my perspective on the topic. And I'm going to sort of speak broadly about reproducibility in science. The National Center for Advancing Translational Science, we spend a lot of time thinking about why drug development fails and reproducibility. And the quality of the science during discovery phase is a really important part of that. And given this problem, I'm going to talk a little bit about the Assay Guidance Manual and a program called ring testing that we're doing between our European and American research partners to try and understand the robustness and reproducibility of high-throughput screening. So here at NCATS, like other centers that you'll hear about later on, we're a translational science center and a small molecule development center. The primary goal of my branch is to develop small molecule probes that are high quality, specific, and high activity against new targets. And to do that, you need a few different fundamental pieces. You need assay development and assay biology programs. You usually need automation and high throughput screening so that you can test many compounds. And you need medicinal chemistry so that you can take the hits from your high throughput screening and optimize them into small molecules that can be used as biological tools, both in vitro and ultimately in vivo. One of the key aspects of the way that we work is that everything we do is a collaboration. So nearly every project we have, somebody comes to us as a collaborator and they're a domain expert in a specific area of biology, and they'll say, I have an idea about a target or a phenotype, and I think if we could modulate this, we might be able to cure a disease or at least study the biology of a disease. And they come to us because they're science experts in a specific topic, but they're not translational scientists. And so they normally have an assay that they've developed for their target. It might not be amenable in terms of scale. It might not even be optimal as an assay. It tends to be a very affordable format. And our job is to transfer that assay over to our branch and test it, miniaturize it, and evaluate the reproducibility of that assay. So does it actually work? Is it an assay? And ultimately scale it down to work in 50, 36 more plates so we can start on the pathway I spoke about in the previous slide. So there's lots of unique challenges from a translational science and a discovery science point of view, but a big one of those is just reproducibility. And at the other end of this sort of pipeline where assay development and screening starts is the private sector, for example, such as pharma, who want to develop drugs. And most of the findings that they act on to start developing drugs come from the people listening to this webinar. It comes from the literature. They read the papers that we read, and if people are writing a paper that says this gene is responsible for this disease, they think, well, this is interesting and it might be actionable and there might be a therapeutic opportunity here. And this is where the big problem comes in, and there are lots of papers that talk about this. I've just got one example here. The challenge we face and that the drug development community faces is that approximately or more than half of all research that's published in the biomedical space is not reproducible. So that's about, in United States dollars, almost $30 billion a year in research funding goes towards publications and science that can't be reproduced by other labs. I know someone on the board of Eli Lilly, and they've told me that, in their opinion, over 3/4 of papers and studies that they try to reproduce as part of evaluating an idea, they fail to do so. So this is sometimes called the reproducibility crisis. There are lots of reasons at a high level that this occurs: poor training, provocative statements. So I think we've all written a paper where we study something in biology, and at the end we say, and so this could be a cure for a disease. And so those kind of extrapolations are read by others in the scientific community and acted upon. Protocols that are poorly written so that they can't be replicated, or just over-interpretation, over-statistical analysis. And so there's a real challenge and a real responsibility for all of us as biomedical scientists to think about reproducibility, to think about the data we publish, and to make sure that the findings that we publish and that we report on are robust and that they're real. Because ultimately, when we're writing a paper, you need to remember someone else might be trying to reproduce that. I just put this slide here. The National Academies for Sciences, Engineering and Medicine wrote a report recently that you could find by searching, and they make a key point here. So technically, reproducibility actually means analyzing the same data and drawing the same conclusions, which is very different from replicability, which is, I think, what us biologists think about a lot, which is where multiple people do the same experiment in different places and can get the same result. So there's actually a difference between reproducibility and replicability when you're talking about biomedical research. So in terms of high throughput screening and that early stage discovery, I just want to give you a few examples that are quite diverse, but give you a sense of just how complicated and how challenging science is, and how we always need to remain alert to all of the various pitfalls that we're coming across and doing high quality discovery work. So the first one that I'm not going to go through in detail, but when you're developing an assay for high throughput screening, there are some really important aspects of this that are essential to make sure you believe in the discoveries that you're making. For example, from an assay development and high throughput screening point of view, we have positive and negative controls, as do at most screening centers, on every single plate from a high throughput screen. So if we screen 250 plates, every plate has a positive and negative control, because we live and die by the statistical power of our plates. If one plate has bad assay statistics from analyzing positive and negative control data on that plate, we throw it away and we repeat it. But beyond that, you want to make sure you have an assay that has a realistic hit rate, so that you believe in the molecules that you're identifying, and that you're not, if you have a 10% hit rate, most of those compounds are probably not reliable and real. You want to make sure after you do a screen that you characterize and understand the quality of the hits in that high throughput screen. And I'll give you an example of that in a moment. And you want your reagents to be stable. And again, all of this goes down to the time and energy spent developing an assay for high throughput screening. It might take 2 or 3 days to run a high throughput screen. It might take a year to develop an excellent assay. And that's where the important work goes in. So I'm going to give you 2 examples of how important understanding molecules and the behavior of molecules is, given that that's what we're ultimately striving to find. So the first one is from a few years ago, but my background is actually studying platinum drugs as chemotherapeutics. And this is platinum here. It's the world's most used cancer drug. And most people who work on platinum and who are experts know that if you dissolve it in DMSO, it will bind to platinum and inactivate the drug. The problem is that in nearly every cancer lab and most high throughput screening labs, if you give someone a drug and say, dissolve this for me and test it, they're going to dissolve it in DMSO. It's the universal solvent that all biomedical scientists use. So my problem was that I received and have a collection of about 1,000 platinum drugs from a company that we wanted to test. And I had this problem about DMSO. I'd always been told it was bad, but I didn't know how bad it was. And so I did this experiment where I took, say, cisplatin from the clinic, from one of my colleagues, and I did a cell killing curve, and you can see it kills cells. But when I dissolved cisplatin in DMSO, you can see it lost all its cell killing activity. So this is a really, really strong phenotype. And so when I read papers and was reading papers about platinum, I'd regularly find that they were dissolving it in DMSO. So I had a few students read actually hundreds of papers, and we just asked the question, in those papers, what do people say in the methods that they dissolve their platinum drug in? And so we read 35 papers from each of these journals, and this is just an example here. So you can see, for example, there are plenty of papers that are dissolving the drug in things like saline or clinical formulation, which is absolutely perfect. The problem was, though, that about 1/3 of papers that we read were dissolved in DMSO. And almost half of the papers that we read in this journal didn't say what they dissolved cisplatin in at all, and most of those were probably DMSO as well. And this was true across all the journals. And you might think, okay, well, that's not great, but I just remind you that those people publishing those papers are studying inactivated cisplatin. They're not studying the drug appropriately. They're not using it in the right solvent. And this is really serious if you think about the fact that there are about 100,000 papers that study cisplatin. So if 1/3 of them are in DMSO, does that mean 1/3 of them are wrong? But and that might seem like a grand statement, but when you think about the challenges of other people taking those papers and trying to reproduce their results, you can see the kind of challenges that we face in science about making sure that we understand our test agents, that we understand the nature of the protocols that other people are executing in the scientific literature. So I published a paper about this called Say No to DMSO, which is only true for platinum. You have to read the paper. But as I said, this is the world's most used class of platinum drugs. And so this is one example I want to give you where understanding the drug you're using and the limitations of that as a test experimental therapeutic are really critical for reproducibility. Another broader challenge in small molecule discovery are molecules called PAINs. So PAINs stands for pan-assay interference compounds. So these are compounds, when you do a high throughput screen, these are the kind of chemotypes that scientists will often find. And in fact, if you read Journal of Medicinal Chemistry every week, you're going to see compounds like this in those journals. But for different reasons, these compounds are poor quality compounds that are reactive and tend to hit in a lot of assays. They're redox active and they can redox cycle, they have color, they're fluorescent, they interfere with assays. And as a result of interfering with assays, they tend to look like really good hits in a high throughput screen, but they're actually terrible molecules. And training and understanding of PAINs molecules is really critical from a high throughput screening point of view. So one of the important things, just as an example for anticipating this, is that when you're executing high throughput screens, you have to design counter assays to ensure that the hits from your high throughput screen are real, and they're the ones that you want to focus on. Counter assays help you weed out all of the molecules that are bad actors, and this is one example of that. So I mentioned that these pains molecules can redox cycle, and so a redox cycling molecule that's in an assay buffer, if you have reductants present, for example, I'm not going to talk you through this chemistry, but can go through oxidation and reduction and produce hydrogen peroxide. So that hydrogen peroxide can react with the protein. It can look like an inhibitor, but actually it's not an inhibitor of an enzyme. It's producing hydrogen peroxide that's just wrecking all of the assay reagents. And you'd be amazed how common this is. And so when you do a high throughput screen, the first thing we do with every hit from every high throughput screen assay is we test it in this assay called Amplex Red. So this is a really great positive control compound. So we call it a great positive control. It's a terrible compound. It redox cycles. It hits in every single high throughput screen that we do. As I said, it does that by reacting with thiols and producing hydrogen peroxide. So in this really simple assay called Amplex Red, now I've got the reference down the bottom, you simply take the dye Amplex Red, you add horseradish peroxidase, and if your compound produces hydrogen peroxide, together with the horseradish peroxidase, they'll convert this dye to a bright red color. And so you can see that on this assay plate here, where purple is being converted to red, and in fluorescence, this is a strongly fluorescent compound. So every compound that redox cycles produces fluorescence, and we know that's a bad compound, and we throw it out. We ignore it through our subsequent high throughput screen follow-up work. So this is a really important and useful assay. And I just want to give you one example here of a typical screening project where we're studying a cancer target. It's a methyltransferase called NSD2. It's implicated in cancer, and it takes a methyl group from SAM and it transfers it onto a lysine tail. So this is a really interesting enzyme. People care about it. There are no inhibitors, and so we wanted to find one. And this is just a pilot screen where we screen about 17,000 compounds. And you can see that after the screen, we picked 289 compounds. We confirm those in the primary assay. We use counter assays to make sure it's not interfering with our luciferase reporter. And we added the Amplex Red assay. And so 2 examples down here of hits are shown. So this first molecule is actually a molecule that was a known positive control inhibitor. It's not a drug-like molecule, but it's a useful tool molecule. And you can see it inhibited our enzyme very, very nicely, and it didn't inhibit any of our counter assays. So this is a nice biochemical inhibitor. This molecule was in our high throughput screening library, and it hit. It looks like a high throughput screen hit. It inhibits the enzyme in multiple different enzyme assays that we have. And so you can imagine somebody taking this molecule and thinking they found an NSD2 inhibitor. But if you look at the Amplex Red assay, you can see the strong gain in signal that we get from this molecule that tells us that it's a redox cycling molecule, and the inhibition of the enzyme that it's causing is non-specific. And so we can throw this molecule away. Whereas for this molecule that doesn't activate the Amplex Red assay, it looks like a biochemical inhibitor. It doesn't inhibit their counter assays. It works on our orthogonal assay. We actually had a second methyltransferase biochemical assay using a different readout, and it looks better. In pharma, more importantly, in our sector as well, when you're doing a high throughput screen, after you've run counter assays to throw out compounds that we call bad actors, a really preferred way to follow up on them is using a biophysical technique like surface plasmon resonance. The reason that's valued is because what we can do using a technique such as this is we can increase the concentration of compound and demonstrate it biophysically binding to the protein. So it's not a measure of inhibition, it's simply a measure of association or binding. But that binding is very reassuring. When we take a bad compound like this, you don't get binding by surface plasmon resonance because it's not binding to the protein to inhibit it. It's producing redox molecules like peroxide and non-specifically inhibiting the small molecule. So this is a great example, and you can read this paper. It came out in JBC last year, where you can see the kind of assays that are needed for discovery, the orthogonal assays that are needed for confirmation, and the counter assays that are needed to exclude bad acting compounds. And you really need to be methodical and develop multiple assays and go through this cascade in a high throughput screening program so that you identify small molecules that you really believe in that you can take forward to medicinal chemistry, because medchem takes a lot of time, it takes a lot of money, and you don't want to find out 2 years later you've been studying a lousy compound that is basically useless to the scientific community. So the 2 examples I just gave you are small molecule examples, but I'm not just blaming molecules for everything. And so I've got one slide here where I want to comment on another really important challenge, especially for cell-based assays in discovery biology. And that's keeping your cell culture clean. And cell culture quality is absolutely critical in discovery. So when I got to NCATS about four or 5 years ago, I started a program where every single cell line that's being cultured in all of the labs in our institute, every Friday, the scientists come and bring media from their labs and we test them for mycoplasma. So mycoplasma are small bacteria. They don't have a cell wall. And the main place they live is in your mouth. And so when you're talking to your friends doing cell culture, you're spraying mycoplasma out of your mouth that lands in cell culture, and it can contaminate and live with cell lines. It was identified over 60 years ago now, and has been a problem ever since in immortalized cell culture. And so we have a policy now where every single cell line people are growing has to be tested when we receive it from a collaborator. It has to be tested once a month when it's in culture, and it has to be tested again before we go into high-throughput screening, because mycoplasma and bacterial contamination in cells can actually really change the phenotype of a cell. And so you can actually identify phenotypes from small molecules that only happen when mycoplasma cells are contaminated by mycoplasma. So again, poor cell culture quality, poor discovery that's not going to be reproducible in another lab where they're using mycoplasma-free cell lines. And more importantly, you're making discoveries that aren't relevant to the biology that you're interested in because of this contamination. It's actually really affordable to do, and it can save you a huge amount, as I said, of time and money, because when you find out you've done a high-throughput screen with a mycoplasma-contaminated cell line, you've got to throw it away and you have to start all over again. The other aspect of what we've been doing is we started implementing STR, so short tandem repeat evaluation is a unique genetic signature, just like humans. The cell lines that we create from humans can also be genetically distinguished and identified. And so we send off the cell lines that we're testing for STR determination to make sure that we haven't switched two cell lines accidentally or contaminated one cell line with another. And again, it's pretty obvious what the negative outcomes of switching cell lines are in terms of a high-throughput screen, and you'd be amazed how readily it occurs. We've received multiple cell lines from our collaborators where they've told us they're studying pancreatic cancer with a special reporter engineered into their cell lines. We do STR and we find out it's a breast cancer cell line. And so you can imagine those kind of mistakes being calamitous from the point of view of making a discovery, but that paper that ultimately says gene X is important in pancreatic cancer, when it's not, it's breast cancer. And all of those people that go and study that gene in pancreatic cancer failed to reproduce it for a very good reason. The initial study was flawed because of contaminated cell lines. So quality in tissue culture is absolutely critical. So all these points I'm making are really important, and the three I've given you are just three of hundreds of examples, thousands of examples I could give you of where people have identified these problems. So what we did at NCATS about 10 years ago is we created a platform called the Assay Guidance Manual. And it literally is a manual. I'll tell you a little bit more about it, and there's a link down here. This is a number of programs, but primarily an online manual of chapters that really give and transfer excellence in assay design concepts, definitions, validated methodologies, and statistical thinking. And you might remember at the beginning I said that the most important thing about high-throughput screening is having a statistically robust assay that you can believe in. And that's because when you do a high-throughput screen, all you're trying to do from hundreds of thousands of molecules is discover statistical outliers. Those are your hits. So the statistical power of your assay is critical. And so we share statistical thinking and assay design and biologically and pharmacologically relevant assay design. And we've got an editorial board, and we seek and hunt down authors from pharma, from the academic drug discovery sector, who are experts in chemistry, biology, informatics, all of the various aspects of translational science, to put together these chapters to share excellence. And the goal of this is that we make this available freely to everyone. So if you're developing an assay, you can read our chapters, you can read from experts about how they develop assays for high-throughput screening, how they follow up on high-throughput screens, how they develop a discovery pipeline, and to ensure that people are trying to practice the state-of-the-art in discovery biology, and to make sure that the science that people are practicing, the science that people are sharing and disseminating is robust, it's replicable, and it's reproducible. And so if you go there, what you'll find is that right now we're at almost 1,500 printed pages of 46 chapters, and we're expanding it constantly. Our goal at the moment is to add about 5 to 10 chapters a year. And you can see the table of content spans from in vitro and in biochemical and cell-based assays, assay artifacts and interferences. We've got lots of chapters on that. As new technologies come around, we're starting to cover those as well, instrumentation, and we're starting to write educational chapters on pharmacokinetics and the whole drug development program. The AGM itself is widely read, as I said. People can learn about all of these different areas that we've shared. We now have almost, this year, we think we're going to get to 500,000 readers per year from all across the globe. And it's always pleasing to talk to someone at a conference and find out that they've read the AGM. We also organize workshops. This is just a small list of some of them that we've organized here. They're mainly in the United States, but we're hoping to organize one in Europe as well next year. They're all free. So whenever we host an AGM, they're posted on our website. And people can come to the 1 or 2 day workshop and attend for free if you can get there. And so next year, I know we're hoping to have one in Switzerland. We're going to have one here at NIH. We're going to have one in San Diego. And we're going to have one in Atlanta, Georgia. And if anyone's interested in hosting an assay guidance workshop, we're happy to work with people to organize them as well. The good news is that if you can't fly to get to a workshop, if you go to this link, and I know all of these slides are going to be shared with you afterwards, you can watch video casts recorded of every presentation from our workshops. And so this is an excellent supplement to the manual itself. The presentations, as you can imagine, are very different to the chapters, and it's another opportunity to learn about the Assay Guidance Manual. We also create workshops around specific problems. I think many of you in Europe are also aware of the opioid crisis. The United States has started putting in a lot of money to studying challenges of addiction and of pain medications. And we organized a focused workshop on assays and discovery challenges for pain and addiction targets. And again, you can watch 2 full days of videocast focused on these topics. So I point you again to the links down the bottom. So the final piece I wanted to mention is that there are lots of experiments in reproducibility that are really difficult to do and that no one ever does. And one of them is high-throughput screening. No one ever does the same high-throughput screen in 2 places. And so we don't really know a lot about how robust a high-throughput screen is. If I do a screen in America and you do one in the Czech Republic, will we get the same hits from the library? You'd like to think we'd find exactly the same things. That would be a sign of an excellent discovery program. But we don't know if that's true. So there's a wonderful program, an initiative called Translation Together, which has members including AATRIS and the NCATS. And we decided to get together and create something called the retesting platform, where we decided to create a couple of biochemical assays with detailed protocols, share them to high-throughput screening centers all over the world that would do the same screen, and then we'd compare our results. And so I'm just going to give you a very brief example of that in the next minute or two. And so what we decided to do is pick 2 very common biochemical assays. They're both bioluminescence assays. One, we use the firefly luciferase. The other, we use the Ranella luciferase. These are very common enzymes used in discovery biology. We created a very detailed protocol, an assay kit that had everything that a lab would need. And we actually shared a library that we created of about 1,500 compounds that we gave to those labs. Same assay, same protocol, same library. Do we get the same results? And we had a phase 0 where Pfizer and NCATS and Eli Lilly and IMTM in the Czech Republic, one of the AATRIS members, took this kit and they tested it. And at a high level, the good news is that from that library, you can see the hit rate was very similar in terms of very active and moderately active compounds. Most were inactive, and that's what you would expect in a high-throughput screening library. And more importantly, some of the really potent molecules that we actually cheated, we added them in anonymously into the library, so the screeners didn't know what they were. You can see that the dose response curves in 4 different screening centers on 2 different contents were very, very similar. So we've now launched into a second phase of this, the full phase, where we've actually shared the kit with a large number of screening centers across Europe, and we're really excited to receive this data now. A couple of centers in Australia. We're comparing the screening data. There's obviously lots of learning moments in this. No one's ever tried to get multiple places to execute the same high-throughput screening protocol. And so there are lots of technical elements of trying to do this, but we're really excited about this retesting program. And it shows that, you really need to study and understand the science of discovery to understand the pitfalls and the challenges for reproducibility. And so I just wanted to give you this really highly collaborative example of the kind of things we need to do to study and understand drug discovery. So this is my last slide, and just, I guess, as some takeaway points, what I'd say here is that when you're developing assays and studying biology, you really need to think rigorously about your assay. What artifacts might arise? What counterassays will you need to identify compounds that are acting artifactually and that are not giving you the activity that you're truly seeking? Are your assays statistically powerful enough that you believe in the molecules that you're identifying? And when you're publishing and sharing, are your methods and protocols detailed enough that they can be repeated? I think that's probably the most critical question of all, in a way. And really the litmus test is, if you asked a colleague to take your assay and go down the corridor and reproduce your data, would they get the same result? And could you look someone in the eye after they've done it and believe in your result? So those are some points for reflection and consideration. And I just want to thank you again for listening. And the link here for the Assay Guidance Manual is there again, and I'm happy to answer questions at the end.
Thank you very much, Matt, for this contribution from the NIH NCATS lab in Bethesda. I will now hand over to the second and the third speaker, Wolfgang and Martin, who are in the lab in Berlin. So, Wolfgang, the floor is yours.
Yes, so thank you very much, also Matt, for this very interesting and informative presentation. So now I'll tell you a little bit about the OPENSCREEN, which is a fairly new infrastructure here developed in Europe. And then in the second part of our talk, my colleague Martin here from one of our partner sites will go to some real-life examples and show how we actually measure and quantify also our high-throughput screening data. EU-OPENSCREEN was just founded about a year ago. It's one of several infrastructures, and our really focus is chemical biology. So just to tell you just a few words, what do we understand about chemical biology? That's for us really the goal of using small molecules to understand biological processes. And in that way, it's kind of complementary to genetic approaches, which were done for many years now. Just knocking out genes or reducing expression of protein levels is relatively simple now, as is done all over the world and in many academic groups. The problem with that approach is that sometimes you get also artifacts. You change not only your desired enzyme, but you change a lot of signaling pathways related to that particular protein. So what you then actually get as a phenotypic readout is often very different to what you expected. And the danger there is that you might draw wrong conclusions from it. So for that reason, we are really investing into new chemical probes. So really small molecules, which are then complementary to the genetic approaches, and then really should help in identifying the role, really the physiological role of certain proteins, signaling pathways in organisms. So I will talk a little bit now about EU-OPENSCREEN, how it was founded and why, and what we can actually offer to scientists. And then really try and highlight the potential for achieving reproducible data, especially in high-throughput screening, and also ensuring quality in high-throughput screening by adapting many of the approaches and the issues, also considering the issues which Matt highlighted in his talk. So, as you know, chemical probes can also be used as drugs, and this is a very old example. The chemical probe, aspirin, is an inhibitor of an enzyme, cyclooxygenase, many years old. It's a kind of chemical probe, but the problem with that molecule is that first of all, it's fairly weak, and also secondly, it's not really selective. It acts also upon other enzymes, for example, COX-2. And it's also not really a competitive inhibitor. It is a covalent binder of that protein. So this would be an example of a bad probe. And what we are really trying to get are molecules of high potency and, more important for us, of very good selectivity. So that's the mission of the OPENSCREEN. And I'll tell you now how we are trying to do this. The OPENSCREEN is one of several life sciences related infrastructures created during the last years in Europe, and we are working together also with others, which you can see on the slide. One, for example, is called Instruct, which is based in Oxford and is really focused on structural biology, and there are others like IATRIS, of course, focus on preclinical research, ICAN on clinical research, and so on. So there are several ones around now, and we're trying to really work closely together with them, with the scientists who do projects with us. So as I said, our focus is on chemical biology. We are now funded currently by 8 member countries. We have about, well, more than 20 partners in these 8 countries. And most of these partners are assay development and high throughput screening centers, academic centers. We have 5 centers more focused on the optimization of the initial hits, so really with expertise in medicinal chemistry. We have a database site, and this is a very important tool. I'll come back to that a little bit later, where we will publish all the structures of all the molecules, but also the experimental results of all our screens, plus protocols and other metadata. So our headquarters is in Berlin, and here in Berlin we really do the compound management. And with compound management we mean just really a fully automated system with a capacity of more than 400,000 tubes for our compound collections, plus liquid handling workstations. And our job here is then to send copies of our library to our screening centers for the actual biological screening work, and then the data will be uploaded in our database. So we have a set of different compound libraries, and the whole concept of the OpenScreen is also shown here on this slide. So we try to provide services for both biologists, pharmacologists, chemists, and database users. We're currently collecting about 100,000 commercial compounds, where we really look into novel compounds, fairly novel compounds, not always showing up in other compound libraries which you might find in pharmaceutical companies, where we really had a strong emphasis on looking at diversity in these compound sets, where we work together with different computational chemistry groups in our member countries to really select these compounds. In addition to these commercial ones, we are collecting over the years from now on basically academic compounds from many academic chemistry labs all over the world. And they might have different, more interesting properties than the ones you would normally find from the usual commercial vendors. So the biological screening at our sites is done according to industry standards. Many of the scientists working in these institutes have formerly worked in pharmaceutical companies. And something which Matt highlighted, of course, is the NIH Assay Guidance Manual, which is now, I think, commonly accepted. And we will set up our own criteria, which are very much aligned to these already existing criteria. So what we also offer is training for scientists to really do high-throughput screening and high-throughput data analysis. As I said, the other important part of our infrastructure is really our open access database, where we keep all the bioactivity data, but also all the physical chemical data on all our compounds, including solubility or assay interference, fluorescence quenching, aggregation potential, these kind of things. Also basic cytotoxicity data on some compounds, which should really help to make sense of our data from coming out from the high throughput screens. So the European Chemical Biology Database, or ECBD, is currently in development, will be finished about mid next year, and it will be open access to everybody, so all over the world. So it's important also to understand here for this database that we will not only keep the positive data, but especially also the negative data, where we then hope that could be the basis also for developing novel algorithms for predicting activity against also novel targets. All compounds in our own collection, which we host here in Berlin, will show up in this database. However, there will be an initial embargo period so that scientists can have the chance to generate IP and publish their data. But really the most important part is that this database will contain everything which is necessary to know about a certain screen, about an assay which was done, so that really people can reproduce these data in their own labs according to the so-called FAIR principles, and FAIR stands to findable, accessible, interoperable, and especially reproducible data, which Matt also highlighted is very important. So in a summary, we really now have, or will very soon have in a few weeks, provide access to our compound sets, to big high-throughput screening sets, but also to smaller pilot library sets of known bioactive compounds. All the screening is done in our screening sites, in our network of partners, while the assay optimization can be done also at our medicinal chemistry sites. So if you want to know a little bit more, of course, you can always contact me or go to on our website, read some of the articles we published last year about this concept of open access chemical biology. And I will now hand over to Martin, my colleague here from the FMP, one of our 17 partner sites, who will tell you a little bit more about how that is done in real life at the FMP.
So hello, I would just like to show quickly what we do as a very basic quality control in high-throughput screening at our site. So what we, for example, do is we create from our values when we measure a plate, a so-called heat map. I think it's pretty much standard. You see here the yellow signals are background, and we have here the last two columns with control samples. And we use this, for example, to detect artifacts like problems with our dispensers. A second piece of quality control is the quality control chart, where we basically sort the plate data after time when it was recorded and calculate the average signals. And here we can see that we, for example, see clearly that we have measured several batches, always a batch starting with high signal and then going a little bit to lower signal. And also one of the things we control is when we, after normalization of the data, we check whether we have something that is similar to a normal distribution, or whether we have two peaks, wouldn't be so good. And of course, we calculate Z prime factor. And this is pretty much standard and very well described in the literature. We just basically adapted it with our software. But there was a thing where we had for a long time a little bit problems, and that was how to basically judge reproducibility or repeatability. And when I looked at the Assay Guidance Manual, there were three methods mentioned: Lin's correlation coefficient, the Blend-Altman method, and the Pearson correlation coefficient. And here we see, for example, on real data, where we measured a plate in two technical replicates. We have measured one plate where we find quite a few hits, and then we can calculate the correlation coefficient, and it's pretty high with about 0.8. We have exactly the same assay. We make the two measurements, but now we have a plate with few hits or very weak hits, and we see that the correlation coefficient suddenly breaks down, and somehow this Pearson correlation coefficient is not a very suitable method to assess reproducibility. Because the reason is it only really works when you have a wide range of measured values, which is only the case if you have really hits on the plate. And we then now switched to the Blend-Altman method to evaluate the agreement between two measurements. And the basic idea is to take the normalized value of each plate from X to Y to calculate the difference of the two measurements and plotting them against the mean of the two measurements as kind of the true value. And then you get plots like this, which tell you over the measured range of values. Basically gives you the limits, the 90% confidence limit, where the differences of the measurements fall into. And so what we do with this method, we take a part of our library, usually the ones with the FDA approved drugs, it's 9 compound model plates, and only measure this part in 2 technical replicates, normalize the data, calculate the percent activities, and generate this Blend-Altman plot. Then you can see here an example. We have here in red the negative controls, in blue the positive controls, and black are the samples. And we can see that we have a nice separation between the control samples, and we also have active samples which fall within the limits of agreement, so that percent activities were basically the same within the two measurements. And we also have here some kind on the diagonal. You see that we have maybe in one of the plates some kind of measurement artifact. And what we use, basically calculate here, is the distance of these limits of agreement. And we found it quite useful, besides the set prime value, so not only to optimize for a large signal separation, but also to use it when you sometimes do not really know which measurement value is best to take. For example, if I have a luminescence measurement, the measurement instruments sometimes give you the raw data, and sometimes, because the readers have some positional artifacts, they give you positional corrected values, where they give you basically a bit nicer heat maps, but we figured out with these plots that the data looks nicer on the heat maps, but it's not so well reproducible. In scope of EU-OPENSCREEN, we will also use the blend-Altman method for the increased library size that we are going to have. It will be 140,000 compounds, and with the usual hit rates of 1 or 2%, we will almost in every screen have to introduce a so-called repeatability test to remove the random false positives. And there is also the question, how do we evaluate this data? We will do that again with the blend-Altman method. So we can see here, when we do a primary screen in one replicate, we should get, after Z-score normalization, a normal distribution. So we have the normal distribution as a model. When we do IC50 measurements, we have the dose response model we can fit to the data. But what do we do if we have only 2 replicates and our data is not normally distributed anymore because we have concentrated the active samples? And that's now an example from real life data where we had this repeatability step with an enriched population which is enriched in actives. And we see here basically in the blend-Altman plot that we have the red population, which are the inhibitors, which repeatedly showed their activity, blue population with activators, and a black population, which showed to be repeatedly inactive. And we basically throw out these compounds and discard them and only continue with the active samples. So, so we found this blend-Altman method very useful for our purposes. And the blend-Altman method is also hidden in the so-called minimum significant ratio, where you do exactly this with IC50 data from different replicates, but it can also be used in primary screening. Thank you very much.
Okay.
Thank you very much to all 3 speakers. Thank you, Matt, thank you, Wolfgang and Martin, for your excellent presentations. We have now some time to address any questions, so please feel free to add questions to the question box. We have one question here, and this is a question for Matt. We are doing a lot of collaborative research. So what do you do in case the results of your collaborator cannot be reproduced? Do you have any advice?
Yeah, so that's not uncommon, and it could be sometimes. We've definitely had the experience that we can't repeat the basic finding. Sometimes we just can't, in inverted commas, make the assay work. But I think in every single case, being open and having a conversation early during reproducibility studies is clear. We always tell potential collaborators that we reproduce their science. They're usually comfortable with that idea. And then as we have problems, usually as we talk through the protocol and share our method with them, they can identify points where we're getting it wrong to fix it. So often a conversation will highlight issues with the protocols, and we get to the point where we can reproduce their finding. Sometimes we can't at all. It's definitely happened. And you can imagine some scientists and their groups can take that quite personally. Although I would argue that if we haven't been able to reproduce it, of course, there are other labs that are going to have the same problem. But being open and honest about that is key. I would say the challenge is, what do you do when you can't reproduce other people's work? And how do you tell the community? And I think that's something all of us as scientists really wrestle with. And at the moment, those things are conversations between us and a collaborating lab, and it's up to them to ultimately address the challenges of the reproducibility of their science.
Okay, thank you very much. I have another question. This is one for Wolfgang. So this is about the EU OPENSCREEN infrastructure. Can you tell us a little bit about who can use the infrastructure and what does it cost?
So the infrastructure can really be used by everybody in the world, but there is a different cost structure, simply because we wanted to make it attractive for countries to join our consortium. So all the scientists from our member states can use our compound collection for a much reduced price. So of course we are a non-for-profit organization, so we cannot make any profit on anything we are doing, but we have to get the money back from the original compounds we have to buy. And our current model looks like that for each compound which is tested in a high-throughput screen, it would cost the scientists about 20 cents. So if you screen 100,000 compounds, the campaign would cost 20,000 euros, which includes also the validation of the hits, the concentration response curves, the counter screens, and these kind of tests. But if someone from outside our member state comes and is interested in the work, and working together with one of our partners, then they would have to pay the double. And the same is true for scientists from companies. So from small medium enterprises, for instance, they also have to pay about 40 cents per compound.
Okay, thank you very much. I have one more question for Martin. What software packages do you use for doing the analysis of your work?
No, we basically switched to freeware completely, and we now use the Constance Information Miner, which is a workflow software, which then also can integrate the R statistics packages for doing the plots and the reports.
Okay. Given the time, I think we will leave it with that. If there are more questions, we can always address them by email, and we will refer to the speakers. So we have now come to the end of this webinar, and we would like to thank you all for attending today's webinar, with a special thanks, of course, to the lectures of the excellent presentations by the speakers. Should you have any further of these questions, we will address them, of course. The recording of this webinar will be made available to anyone interested on our website within the next 2 weeks. And finally, we would like to point out that there will be another webinar in 3 weeks' time, and this is about reproducibility in preclinical and animal experimentation. So we hope to all see you again then, and thank you, and have a nice day.